Hello everyone, I am Jomaribor Hamidel, BSc2 Alpha, and in this video, I am going to discuss the salient points of each topic from Lesson 1 to 4, which is the coverage of the midterm examination. Okay, so let's start. Lesson 1, Introduction to Forensic Chemistry. Lesson 1.1, Development of Scientific Crime Laboratories. What is forensic chemistry? Forensic chemistry is the application of chemistry to criminal investigation. It focuses on the chemical analysis of substances connected to a crime. Forensic, etymologically, in Latin means of or before forum. Collection or reception of physical evidence to be examined. Evidence collection is the crucial knowledge that may help incident responders in understanding the process of attack and tracing the attacker. Therefore, the incident responders ought to apprehend where they will find the proof and the way to collect it. A wide variety of physical evidence can be collected at a scene that is deemed valuable or probative for collection and investigation. Number 1. Biological evidence such as blood, body fluids, hair, and other tissues. Number 2. Latent print evidence such as fingerprints, palm prints, and footprints. Number 3. Footwear and tire track evidence. Number 4. Trace evidence such as fibers, soil, vegetation, and glass fragments. Number 5. Digital evidence such as cell phone records, internet logs, email messages. Number 6. Tool and tool mark evidence. Number 7. Drug evidence. Number 8. Firearm evidence. The actual examination of specimen. When collecting specimens for forensic analysis, the following principles should be strictly adhered to. Avoid contamination. Ensure that specimens are not contaminated by other materials. Wear gloves at all times. Modern DNA assay systems are very sensitive and may detect small amounts of extraneous material. Collect early. Try to collect forensic specimens as soon as possible. The likelihood of collecting evidentiary material decreases with the passing of time. Ideally, specimens should be collected within 24 hours of the assault. After 72 hours, yields are reduced considerably. Handle properly. Ensure that specimens are packed, stored, and transported correctly. Analytical laboratories should be able to provide guidance on special requirements for specimen handling and storage. As a general rule, fluids should be refrigerated. Anything else should be kept dry. Label accurately. All specimens must be clearly labeled with the name and date of birth, and type of specimen and the date and time of collection. Ensure security. Specimens should be packed to ensure that they are secure and tamper-proof. Only authorized people should be entrusted with specimens. Maintain continuity. Once a specimen has been collected, its subsequent handling should be recorded. Details of the transfer of the specimen between individuals should be also recorded. It is advisable to check with local authorities regarding the protocols for the recording of such information. Lesson 2. Dangerous drugs. What is dangerous drugs? Drugs are chemicals that affect a person in such a way as to bring about physiological, emotional, or behavioral change. Dangerous drugs are those that have high tendency for abuse and dependency. These substances may be organic or synthetic and pose harm to those who use them. Drug abuse. Drug abuse exists when a person continually uses a drug other than its intended purpose. This continued use can lead to a drug dependence, a state of physical and psychological dependence or both on a dangerous drug. Drug addiction. Drug addiction is a complex and often chronic brain disease. It is characterized by excessive drug craving, seeking, and use. Addiction is caused by brain changes caused by constant drug use. Drug dependency. This is a state of physical and psychological dependence, or both, on a dangerous drug or drugs, experienced by a person following the use of that substance on a periodic or continuous basis. A person dependent on drugs will experience withdrawal reactions, also known as cold turkey symptoms that occur after a long-term use of drug is reduced or stopped abruptly after abstaining from drugs. Key terminologies regarding drugs. Dangerous Drugs Board. The Dangerous Drugs Board DDB leads the campaign against drug abuse, illicit trafficking in the Philippines. It was established in 1972 through Republic Act 6425, composed of only 7 national government agencies as members. Republic Act 9165, an act instituting the Comprehensive Dangerous Drugs Act of 2002, repealing Republic Act No. 6425. Republic Act 6425, otherwise known as the Dangerous Drugs Act of 1972, as amended, providing funds therefore and for other purposes. This act, consisting of 101 sections, provides for importation of dangerous drugs and or controlled precursors and essential chemicals. PDEA, the agency tasked with the enforcement of the penal and regulatory provisions of the Republic Act No. 9165. Depressant drugs. Depressant drugs reduce activity in the central nervous systems. Examples include alcohol, GHB, ketamine, benzodiazepines, opioids. Serotonin syndrome. A life-threatening condition that usually starts within 24 hours of taking the drug. It occurs when the brain is overloaded with a neurotransmitter called serotonin. Symptoms include coma, seizures, shaking, confusion, rigid muscles, rapid heartbeat, and overheating. Stimulant drugs. These increase the activity in the central nervous system. Examples include cocaine, caffeine, MDMA or ecstasy, methamphetamine, speed, ice, crystal meth, base, Ritalin, and some new psychoactive drugs such as mephedrone and BZP. Polydrug use. Mixing drugs or taking one drug when under the influence of another drug is known as polydrug use. Withdrawal effects. People who are dependent on drugs or alcohol may also experience withdrawal effects when they stop using. Withdrawal effects can last for several days to many weeks, depending on the type of drug and severity on their dependence. Withdrawal symptoms may include feelings of anxiety, depression, restlessness, irritability, and aggression. Withdrawal can also cause muscle spasms, headaches, muscle cramps, diarrhea, vomiting, and cravings for the drug. Lesson 2.1. Classification of drugs. Number 1. Central Nervous System Depressants. CNS depressants slow down the operations of the brain and the body. Examples of CNS depressants include alcohol, barbiturates, anti-anxiety tranquilizers, GHB, rohypnol, and many other antidepressants such as Zoloft and Paxil. Number 2. CNS stimulants. CNS stimulants accelerate the heart rate and elevate the blood pressure and speed up or overstimulate the body. Examples of CNS stimulants include cocaine, crack cocaine, amphetamines, and methamphetamine crank. Number 3. Hallucinogens. Hallucinogens cause the user to perceive things differently than they actually are. Examples include LSD, peyote, psilocybin, and MDMA or ecstasy. Number 4. Dissociative anesthetics. Dissociative anesthetics include drugs that inhibit pain by cutting off or dissociating the brain's perception of the pain. PCP, its analogs, and dextromethorphan are examples of dissociative anesthetics. Number 5. Narcotic analgesics. Narcotic analgesics relieve pain, induce euphoria, and create mood changes in the user. Examples of narcotic analgesics include opium, codeine, heroin, demerol, darvon, morphine, methadone, Vicodin, and Oxycontin. Number 6. Inhalants. Inhalants include a wide variety of breathable substances that produce mind-altering results and effects. Examples of inhalants include toluene, plastic cement, paint, gasoline, paint thinners, hairsprays, and various anesthetic case gases. Number 7. Cannabis. Cannabis is a scientific name for marijuana. The active ingredient in cannabis is delta-9 tetrahydrocannabinol or THC. This category includes cannabinoids and synthetics like dronabinol. Classification of drug addicts. Situational users. When a person uses drug to cope with the demands of particular situations. Spree users. One who has illegally, wrongfully, or improperly used drugs for kicks or for the experience. Hardcore addicts. are individuals whose actions almost exclusively center around the drug experience in obtaining supplies. Hippies promoted the recreational use of hallucinogenic drugs, particularly marijuana and LSD, in so-called head trips, justifying the practice as a way of expanding consciousness. Experimenters were those who had used marijuana, but less than 10 times in the past year and less than 3 times in the past month. Casual are drug users who are just trying it once or a few times. Every drug user starts out as an occasional user, and that initial use is a voluntary and controllable decision. Regular, the person using an illegal substance more than that over a weekend. Compulsive, a pattern of drug consumption that is stimulus-bound, stereotyped, difficult to regulate, and identified by a loss of control over intake. Pharmacological classification of drugs. Opium. It is dried latex obtained from the seed of capsules of the opium poppy, Papaver somniferum. Approximately 12% of opium is made up of the analgesic alkaloid morphine, which is processed chemically to produce heroin and other synthetic opioids for medicinal use and for the illegal drug trade. Morphine is a pain medication of the opiate family that is found naturally in a dark brown, resinous form from the poppy plant. It can be taken orally or injected. It acts directly on the central nervous system to induce analgesia and alter perception and emotional response to pain. Codeine is an opiate and prodrug of morphine used to treat pain, coughing, and diarrhea. It is used naturally in the sap of the opium poppy, Papaver somniferum. It is typically used to treat mild to moderate degrees of pain. Heroin is a highly addictive drug made from morphine. It can be injected, snorted, or smoked. Paregoric, or camphorated tincture of opium, also known as Tinctura opi camphrata, is a traditional patent medicine known for its anti-diarrheal, anti-tussive, and analgesic properties. Demerol, or known as meperidine, has a risk for abuse and addiction, which can lead to overdose and death. Meperidine may also cause severe, possibly fatal, breathing problems. Methadone is indicated for the management of pain severe enough to require an opioid analgesic. Barbiturates are a kind of depressant or sedative drug. They are an old class of drug used to relax the body and help people sleep. These drugs were first developed in the late 19th century. Tranquilizer is used to reduce anxiety, fear, tension, agitation, and related states of mental disturbance. Alcohol is one of the world's most popular drugs, but its natural substance, not a careful concocted pharmaceutical. Lesson 3. Semen and Seminal stain. The presence of sperm and semen can be important to a crime investigation. The visual detection of semen can provide evidence in the case of a suspected rape. As well, the genetic material present in the sperm can be analyzed and used as a genetic fingerprint to identify a suspect. In a forensic examination, semen can be detected by the presence of the enzyme acid phosphatase. Key terminologies. Semen is a grayish-white bodily fluid that is secreted by the gonads of male animals. It carries sperm or the spermatozoa and fructose and other enzymes. Spermatozoa are the male sex cells that carry a man's genetic material. Chromosomes is an organized package of DNA found in the nucleus of the cell. Humans have 23 pairs of chromosomes, 22 pairs of numbered chromosomes called autosomes, and 1 pair of sex chromosomes, X and Y. Sperm mitochondria produces energy for the movement of the sperm. The sperm mitochondria, as well as the mitochondria in the some somatic cells, contains its own DNA. Sperm morphology refers to the size and shape and appearance of a man's sperm, which when abnormal can decrease fertility and make it more difficult to fertilize the woman's egg. Lesson 3.2. Examination of semen and seminal stain. Number 1. Physical examination. Ultraviolet examination, bluish fluorescence indicates positive result. Number 2. Chemical examination. Fluorescence test, dark brown rhombic or needle-shaped crystals of choline periodide indicates positive result. Acid phosphate test, purple color indicates positive result. Barbiturates test, purple color indicates positive result. Number 3. Microscopic examination is confirmatory test in forensic serology to identify whether it is semen or not. The experiment carried out by an expert follows a protocol, and according to that experiment takes place, the collected samples are first sealed and sent to the forensic lab. Microscopic examination of semen is confirmatory test in forensic serology to identify whether it is semen or not. The experiment carried out by an expert follows a protocol, and according to that experiment takes place, the collected samples are first sealed and sent to the forensic lab. Sample slides are prepared and examined under microscope. These prepared semen sample slides will be used as a forensic evidence for future investigation. For forensic purposes, the composition of semen can be simplified into two components, seminal fluid and spermatozoa. Microscopically identifying spermatozoa is an absolute indicator that semen is present on an item. Lesson 4. Blood and blood stains. The use of blood in forensic analysis is a method for identifying individuals suspected of committing some kinds of crime. Blood as evidence holds significance in the criminal justice system, as it can link a crime with a criminal or exclude an individual's involvement in a crime. Key terminologies. Blood is a specialized body fluid. It has four main components: plasma, red blood cells, white blood cells, and platelets. Blood stain, a spot or stain made by blood. Blood stain pattern analysis is the field of forensic science that consists of the study and analysis of blood stains at a known or suspected crime scene with the purpose of drawing conclusions about the nature, timing, and other details of the crime. Plasma is a light amber-colored liquid component of blood in which blood cells are absent but contains proteins and other constituents of whole blood in suspension. Platelets are pieces of very large cells in the bone marrow called megakaryocytes. They help form blood clots to slow or stop bleeding and to help wounds heal. Hemoglobin, a red protein responsible for transporting oxygen in the blood of vertebrates. Veins, carries the blood back to your heart to absorb more oxygen. Fibrin, an insoluble protein formed from fibrinogen during the clotting of blood. Lesson 4.1. Blood and the importance of the study of the blood. At the crime scene, it is important to establish the type, origin, and other characteristics of the blood or blood stain. Preliminary investigations in this regard are to confirm whether the recovered fluid is blood and whether its origin is human. Once the human origin of the blood or blood stain is confirmed, further analysis is done to establish other characteristics. The blood stain is initially subjected to blood typing. Proteins, enzymes, and antigens present in the blood of the individual are also analyzed. Composition of blood. Blood is a specialized body fluid and has main components of plasma, red blood cells, white blood cells, hemoglobin, and platelets. Blood has many different functions, including transporting oxygen and nutrients to the lungs and tissues, forming blood clots to prevent excess blood loss, carrying cells and antibodies that fight infection, bringing waste products to the kidneys and liver, which filter and clean the blood, and regulating body temperature. Blood analysis. Forensic serology is the application of immunological and biochemical methods to identify the presence of a body fluid or tissue sample encountered in connection with the investigation of a crime and the possible further genetic characterization of the sample with a view to determining likely donors thereof. Here, the genetic characterization involves polymorphic antigens and the proteins present in the blood and does not include DNA genetic markers. Forensic serology thus is a study and examination of body fluids such as blood, semen, sweat, urine, saliva, fecal matter, and other fluids mainly for identification purposes in criminal medicological investigations. Blood is the most commonly recovered and one of the most important pieces of evidence utilized in forensic investigations. It is commonly recovered in cases of homicide, assault, and terrorist attacks, including bomb blasts. Percentage of blood group among Filipinos. Type A, 26.3%, Type B, 24.7%, Type O 43.3%, and Type AB 5.7%. That's all for today's video. Thank you for listening. God bless.